Thanks, Bilal, for inviting me.
One final reminder for the students and postdocs, there's lunch afterwards, so you can pick your brains afterwards at lunch.
Thanks. Yeah, despite the fact that I grew up in the Midwest, I do not like this weather at all. So this is our Parker Petit building, just to show you that we get some cash from that guy too. So today, I'm going to try to keep things fairly general because I know this is a very diverse audience. And so one of the main things that we're interested in my lab is how the brain can repair itself after damage. And of course, that's related to the mechanisms that it uses during development as well. So we study normal development as well as plasticity and responses that the brain has to injuries or other insults. And of course, as you probably realize, we're all quite good at damaging our heads. So we've heard a lot of news about CTE and football players lately. And by the way, that happens in soccer players too. Soldiers, little kids are always trying to kill themselves. And then we do these crazy sports like that or that. And then even if you don't do any of those things, if you drive in Atlanta, you're doing this. So lots of ways we can injure our heads. The difficulty being that when you go into the hospital with a head injury, basically what they do is they stop the bleeding and then tell you, well, we'll just have to wait and see how things turn out. Don't you think that's a little primitive in this era of modern medicine? So we ought to be able to have a better response to head injuries than waiting for the brain to do most of the work in healing itself. So we're trying to understand how the brain does that so that we could harness those mechanisms and improve therapeutic approaches. So how do we choose a model system for traumatic brain injury? Well, it has to be a system that we can manipulate fairly easily. And in my laboratory, we actually use 3 different model systems. We use the hamster when we're interested in looking at the retinocollicular projection, so from the retina to the visual midbrain. We use ferrets when we want to do cortical physiology, because trying to do cortical physiology in rodents is a bit of a challenge. So we like to avoid that. And then if we're forced into using knockouts and transgenics, then we grudgingly use mice, which are a lot harder to do physiology in. So we use these 3 models, and today I'm going to talk entirely about the hamster and the mice results. So how can we manipulate sensory pathways or alter their development in a way or injure them so that we can study these mechanisms of repair? Well, we could just put a lot of extra input in and see if the brain knows how to accommodate that extra input. That's a question that's more related to evolutionary hypotheses about the brain. But we do sort of increase the input in one of the approaches that I'll show you today, and I'll explain that a little better in a moment. We also can decrease the amount of input. So a large proportion of the lab right now is studying the effects of sensory deprivation. Or we can rewire the inputs. So we can rearrange inputs in relation to their targets, and that's work that I would like to talk about, except I haven't got anything new on that for the last 4 or 5 years. I haven't yet found a student who wants to do the 36-hour recording sessions. Any volunteers? The student that did that work is in Philadelphia now doing her postdoc, Yu-Ting Mao. She was a beast. So you're probably familiar with a slide that looks something like this, showing you how sensory information can be arranged topographically. This is the most common figure that you might see in a medical neuroscience textbook, showing you how the body surface, your skin surface, and the receptors in your skin are represented along your somatosensory cortex. So there is a 2-dimensional representation of somatosensory information in your brain. There's also a 2-dimensional representation of visual information, and that's what we're going to be concentrating on today. In particular, the model that I'm going to talk about was coming out of work really done by Roger Sperry. You may remember him as the split brain patient guy. He won the Nobel Prize for that work. But he did this work as well in the 1960s, where he turned the eye of a frog upside down, cut the optic nerve, and let the optic nerve regenerate, because frogs can do stuff like that. We can't. We'd like to know why not. But at any rate, what Sperry noted was that the optic nerve, even though the eye was upside down, it grew back to the same location in the midbrain where it used to be. So he used the idea of chemical attraction and pre-specified information to develop this chemoaffinity hypothesis. And so as a result, this was a behavioral experiment, so he didn't know what was going on in the brain, but the frog was unable to catch its dinner because if there was a fly here, it would always look for it down there because the eye was upside down, but the connections hadn't changed. Now, what Sperry didn't realize is perhaps because he was one of those type A guys, Nobel Prize winner, and he didn't have the patience to wait to see what the long-term effects of that was. So he had no idea that the frogs would eventually correct that situation because they want to eat. So the brain has a pretty strong motivation to correct its connectivity based on the information that it's getting in its world. And it took John Schmidt, who is a professor at SUNY Albany, who started doing this work and similar manipulations such as the ones I've shown here, to show that the system can regulate itself. So I think I probably can't, well, I don't know, maybe I can. So we see here the retina, and we have what's supposed to be a green, a red, and a black retinal axon coming here. They're making their topographic projection into the optic tectum of, for example, a goldfish or a frog, similar idea. So they each have a particular domain, and then this region in the tectum would be paying attention to the region of visual space that this part of the retina is looking at. What John Schmidt showed was that if you ablate half of the tectum, and this is in an adult because it's a frog or fish, you can do these kinds of things with those guys. You notice now that this green one doesn't have its normal target. The red one's kind of dancing on the edge of not having any place to go. But what happens is that we get a compression of the projection, a compression of the topographic map. So nice that that happens in the so-called lower vertebrates. But in fact, it also, and this was the discovery of my PhD advisor, Barbara Finlay, it also occurs in mammals if we do it early enough. And so we use neonatal hamsters because, as you can see, they're extremely immature at birth. Although apparently they can talk. Just this guy's really got a lot to say here. But they're very immature. It only takes about 15.5 days to make a hamster. So that's a lot faster than most other animals, most other mammals. And so the idea is that we can get access to very early developmental stages compared to what we could do in other animals like cats and primates, which were the model systems of the day in looking at the visual system. And as a result, then, we can get access to the visual projections from the eye before they have made all their connections within the brain. So maybe in this developing system, Barbara Finlay thought we could get this compression to occur, and in fact it does. So just to give you a little orientation here, I'll try to be equal opportunity with the screens here. So here's a general projection from the eye into the visual thalamus, lateral geniculate nucleus, or the superior colliculus, which is the visual midbrain. LGN projects into visual cortex projects back to superior colliculus. So that's just the general anatomical picture here. And if we look at the dorsoventral and nasal temporal axis of the retina, those are re-represented within the tectum to produce that topographic map. So if we were to do an electrophysiology experiment, which is the way we have approached this first experiment I'll tell you about, we can, in an anesthetized hamster or mouse or any other animal we should so choose, but those are the ones we're using, we can put an electrode in the superior colliculus. By the way, superior colliculus, optic tectum, those are the same thing. But it's called superior colliculus in mammals and not in fishes, frogs, reptiles, for some reason that I don't understand. But it's the same thing. So if we were to move our electrode along to different locations in the tectum, we would find that we would get a progressively different location of the visual field that it is representing there. Now, if we remove half of that superior colliculus at birth in these really immature animals, we find the same thing. The map has now scrunched itself down onto the smaller target. Now, originally, this was supposed to produce a situation, this was the plan that Barbara and I had, that this was going to produce a situation where we have the same amount of information in the retina, but it's converging onto a smaller space, so any individual cell within that target seems perfectly logical to conclude that each cell is going to have to represent more visual space. And we were really interested in doing that because there was a huge debate in that day about whether receptive field properties such as orientation, tuning, et cetera, were built from specified parallel pathways or from convergence of different inputs. So that was the big debate, and we were going to answer it. And I was very upset to find, in fact, that even though we're representing twice as much input on half as much target, the individual neurons in the superior colliculus are still looking at the same amount of visual space. So they have repaired themselves to keep the size of receptive fields the same. And of course, I was really upset, not realizing that this was even cooler than what we had planned to have happen in the first place. So graduate students, if you find that you get the opposite of what you expected, do not despair. It can lead to an entire career. So if it had not made that correction, right, the cost of increasing receptive field sizes in a topographically mapped projection would be to alter acuity so that it would look more like that. So you'd have a much fuzzier view of the world. And you might think, well, for a hamster, what difference does it make? But it could, in fact, make a difference. And we're in the process now of doing some behavioral experiments to check their perceptual ability. And it looks like they really are impaired in their escape behavior. So they don't escape as well from a looming stimulus coming from above, which for a hamster, you know, that's likely to be a raptor predator. And they need to be able to escape quickly, because those guys move pretty quickly too. Okay, so that led us then to ask this question, how do they do that, right? How do you get a preservation of receptive field size even though you've got this increase in convergence of the two populations? And so we proposed that perhaps the NMDA receptor, which has some special properties that I'll tell you about, might be able to tell the cells in the superior colliculus how much visual information, that is how much space they were representing, and then be able to prune down the projections enough to keep the receptive field properties the same. So our hypothesis was that NMDA receptor activity is necessary to control the convergence at that level of the single neuron. So it was very important that we make single unit recordings rather than multi-unit or field potential recordings. So we blocked NMDA receptors and compared results in normal animals and animals with the compression manipulation, which I will call see, what did I decide to call that today? Well, you know how you have lab lingo and you have little abbreviations for manipulations you do, and you go talk to an audience and they have no idea what you're talking about. So we could call it lesioned, or we could call it a reduced size superior colliculus. So let's go with that. And so then we're going to assay the receptive field sizes as we did before. Now, just to give you a little bit of background, because it's such a broad audience, we can generally go when we're looking at activity's role in shaping neural connectivity, we can generally go with the maxim that if you use a projection, it's going to get stronger. If you don't use a projection from input to target, it's going to get weaker and you might lose it. And so we can build up our brain power with practice, similarly the way you use your muscles in the gym to build up, or we could have the unfortunate opposite result. And this really happens, apparently, because it happened to Homer. So here's a little diagram to show you what I mean here. So here's our retina, and we have a visual stimulus that's going to move across the retina. That is going to cause firing in a group of neurons that represent that location, and different neurons represent the next location, and so forth. So we'll have a wave of activity traveling across the retina as the stimulus moves from one side to the other. Early in development, you see that any individual target cell can get input from many different retinal axons. And then later on in development, it refines such that it has a smaller receptive field. And the reason that happens is because the target is looking for correlations in activity. So these two are firing together in time. So that leads us to our second maxim of fire together, wire together, that you may have heard in your classes. And so since these two are coincidentally active, they're going to consolidate their connections onto this cell. And likewise, it would lose the connection with that one because it's not as simultaneous, but these two are similarly active and so forth. So the NMDA receptors work, and this may be a review for you, but NMDA receptors work in a number of special fashions. So they're glutamate receptors. You don't have NMDA in your body because that's a drug. That's just the thing that we use to recognize NMDA receptors. But it is a type of glutamate receptor, like the AMPA receptor, which passes sodium, but NMDA receptors pass sodium and calcium. And calcium feeds into second messenger processes that can influence the stability and the strength of that synapse. The other special thing about NMDA receptors is that they are not able to open their pore at the resting potential because there's a magnesium ion that's stuck in there. It's stuck in the pore, and so we can't get sodium and calcium into that channel at resting potential. However, if we can excite this postsynaptic terminal some other way to depolarize the neuron a little bit, now we have some depolarization, the magnesium ion is expelled. Now we can pass calcium and sodium through the NMDA receptor. The calcium leads us to second messenger pathways that can influence the stability of the synapse, to make a long story short. But the idea is that this is a coincidence detector because you have to first one depolarization and then shortly thereafter another one. So just like classical conditioning, when you need to associate an unconditioned stimulus and a conditioned stimulus, this one glutamate receptor allows the postsynaptic neuron to distinguish the level of co-firing of different inputs that are competing for its attention. Okay, so again, we're going to manipulate those NMDA receptors and test the hypothesis that they're necessary for refining the receptive fields after that manipulation. Most of what I'm going to show today is not original data because I want to give you a lot of information, so it's going to be lots of graphs and stuff, so I'll try to not make it too boring. But so here's the process. So we're getting large receptive fields and we're going to refine them down. In the superior colliculus of a hamster or a mouse, the receptive fields of individual neurons are fairly large, much larger than they would be in visual cortex, about 10 degrees in diameter. And as I told you, when we do this manipulation of partially lesioning the tectum, our lab lingo for it is PT, for partial tectum, then the receptive field size is maintained. If we block the NMDA receptors during development with a slow-release polymer that's steadily leaking this antagonist of NMDA receptors called DAPV, then we find that that blocks the normal refinement of the receptive fields. So our receptive fields are bigger by about 50%. If we combine that with the lesion, so we lesion them at birth and we block NMDA receptors, we find, of course, that it blocks the normal refinement, but it also blocks the capacity of the system to compensate for the loss of target tissue when the receptive fields get even larger because of that increased convergence from the retina in the reduced tectum. And then, you always do the control study last, right? Because those are the boring studies. So this is the inactive isomer of the drug, and thank goodness it didn't show anything. So that was an important control with that. So the LAPV, the LAVO form of the drug, isn't active. So a little ball and stick diagram here to illustrate what we've done. This is my idea of a computational neuroscience model. So early in development, we've got a projection from retina into superior colliculus, right? If we record from, this is where I need a fishing pole and not a pointer. If we record from that second neuron in superior colliculus, you're going to see it gets 3 different inputs, right? So its receptive field diameter is going to be 3 different diameters of retinal neurons. Normally, during development, that would refine. Let's say we end up with 2 inputs instead of 3. So that will be the receptive field size of this individual neuron. It'll be sharper in its acuity. What we found in the case where we reduced the tectum by about half, so we went from a 4-cell to a 2-cell tectum in my diagram, we get overlap as we would initially, but the individual axons reduce the size of their arbor, and they'll selectively project to alternative targets so that they have a chance to find target space. So it's kind of like a game of musical chairs with axons. If they don't find a chair, they die. It's a more severe penalty than at the birthday parties. And you notice this has to happen in a very specific way. It still has to select for 2 adjacent inputs. It can't pick that one and that one, or that one and that one, because then the receptive field size would be larger. So there's some way to filter that through, and we proposed that it was due to the NMDA receptor. So we block the NMDA receptors in a normal animal, full-size superior colliculus. We maintain that early state forever. If we combine that with a partial ablation of the tectum, we went from a 4-cell tectum to a 2-cell tectum in this pathetic model here. Then you see that we get an even further increase in receptive field size because we block the normal refinement as well as blocking the compensation for the damage. Okay. So our results then support the hypothesis that NMDA receptor activity is necessary for not only the normal refinement process, which is already known in a number of other systems, but also for compensating for the lesion. So receptive field size is one aspect of the properties of the target cells in superior colliculus, but those neurons also have other special properties that allow the animal to conduct its life. And for a frog, of course, that includes catching moving objects. That was what Sperry's frogs had trouble with. So they need to know something about the objects that they're looking at. They need to have object recognition ability. And so for a frog, that can often just be something as simple as knowing how big and how quickly the target is moving to identify it as a particular kind of prey item that might be tasty. And the same is true of rodents, who will often identify moving objects. So even though we have a maintained receptive field size, maybe we can go back to our original question and address whether convergence from the retina is playing a role in the size and velocity tuning of these neurons. So remember that this is our manipulation. And we're going to ask whether that NMDA receptor blockade combined with the partial lesion of the tectum affects stimulus size and velocity tuning. We thought certainly it would, because we were thinking that target cells, it's not just one target cell that defines direction and velocity, because the stimulus is moving. And so you have to compare from one cell to the next. And from one cell to the next, there's going to be more information from the retina represented. So we thought certainly this is going to change velocity tuning, and it would likely change size tuning too, because of the difference in representation of adjacent target cells. However, to make that story quite short, it had absolutely no effect. So not only was receptive field size maintained after the lesion, although not after the NMDA receptor blockade, but stimulus size and stimulus velocity tuning was resistant not only to the lesion, but it was also resistant to the NMDA receptor blockade. So that anything we could throw at this system, it just fixed itself. Everything just stays the same. So it keeps its acuity, it keeps its size tuning, it keeps its velocity tuning. We were getting a little frustrated that there's just nothing we can do to perturb this system. It is so robust to any kind of manipulation. And on the one hand, that's very interesting, but it wasn't what we were trying to do. So we never did answer that convergence question, but they got into this idea of how inhibitory neurons can often be recruited to compensate for changes in excitatory convergence. So we've got all this extra information coming in from the retina to a given area of the target. This is a little bit hard to explain, so bear with me. But if I were to pass a light stimulus through the center of a receptive field, here's our center right here of a receptive field, I'd get a really strong response. If I moved that stimulus a little to the side, I would eventually not get a response anymore, because I would be outside of the receptive field. Well, we wanted to probe inhibitory connections. And because we're recording extracellularly, we don't know anything about inhibition except how it affects excitation. So if we have excitation and inhibition going on simultaneously, we should see a decrease in excitation from our electrode that's outside of that neuron. So what we do is we take the stimulus that's in the center and we add another stimulus and assay what's called surround inhibition. So superior colliculus cells, unlike cells in the LGN, the thalamus, like really small receptive fields, and they have a lot of inhibition throughout their receptive field. So if we add another stimulus in there, it's going to inhibit the response to the first one. And as we move that second stimulus away from the center of the receptive field, the influence of the inhibition on this response is going to decrease. And in fact, in normal animals, the black lines here, it falls off pretty rapidly, but then it plateaus out. What we found in the animals that had been treated with the NMDA receptor blockade is that the boundaries of inhibition were extended, perhaps to correct for that excess of excitation. And we're proposing, and I won't go into it today, but we built some models when I had an engineering postdoc in the lab. Khalil Razak, who's now a tenured professor at UC Riverside, made some models of how this increased inhibition could fix the stimulus size and velocity tuning, and it worked out really nicely. So we're thinking that this change in inhibitory connectivity is a general means through which plasticity can occur that was previously unappreciated. And most people think about neurons being excitatory toward each other rather than inhibitory, but inhibition shapes responses, and it seems to be very important later on in postnatal development when excitatory connections have already been established. There seems to be some indication from our results and others that inhibitory plasticity is a little more flexible later in life. Okay, so our results were that the inhibition is changing its configuration, and that may compensate for the increased receptive field size and be maintaining size and velocity tuning. So the system can be very flexible in accommodating changes to its inputs and outputs, and that's one way that it preserves function in addition to changing the arbor size of the inputs. Okay, but that didn't tell us, it told us how resistant the system was to manipulation, but it didn't tell us how the compensation process at the very beginning, the compensation for the lesion, was actually happening. And in the meantime, after we did this earlier work, and I started my own lab, and then we started getting more into the system. So I did a postdoc completely out of this area at MIT. I'm not talking about that work today. The ferret work started there. But we wanted to address the molecular basis of this situation. So we know that there are two basic steps in refining the projection from the retina into its target structures. One of those steps is directed by molecular gradients. So the initial formation, the gross topography of the projection is set up based on molecular cues that when we started this work we didn't know what they were, but in the mid-90s they were identified as repulsive axon guidance factors called ephrins, and their receptors, the F receptors, which doesn't start with an F, it's EPH. And so we were reasoning that if you need this gradient of repulsion to set up the initial projection, maybe it's the case that when we injure the tectum, the guidance molecules change their configuration, and they're what tells the map to compress on that smaller fragment. And so we wanted to combine the ablation of the tectum, the partial ablation, with the map compression and assay in a descriptive study for what happens to those ephrins and their receptors, the Fs. And this work was done by a couple postdocs in my lab, Mei Zhu and Qi Cheng, and by a very talented student who was a master's student at the time, pretty much directed this project, Tizuta Tedessa. Now, let me give you some introduction here. So if we have our ephrin-As, and to simplify the story, they're going to be ligands today, although they can actually act as both, which is really irritating. But they're just amazing molecules, very well studied in cancer research. So they are guiding invasion of, for example, blood vessels into tumors. So the ephrin-As are located in the target, and they're from a low anterior to high posterior gradient. Their receptors, which are located in the retina and on the axon terminals, are in the opposite configuration because it's a repulsive interaction. So the high FA receptor expressing region in temporal retina doesn't like the back of the tectum because it's very inhibitory. It can detect how inhibitory that is because the receptor level is high. So it prefers to be here where there's not so much of that ligand. Whereas where the receptor is low, those axons are tolerant of the higher ligand levels. And then intervening steps you can imagine to set up the entire projection. So that was what we knew going in. We did expression studies in hamsters. They'd already been done in mice because we wanted to do hamster physiology and not mouse physiology because it's easier. We found the same thing in the hamsters that we did in the mice. And this is a lateral view of the brain. So this is cortex here that's up, down, nose, tail. Cerebellum there, you may recognize. This is the tectum from there to there. That's the inferior colliculus. Early in development, you see that these yellow areas are showing the mRNA expression pattern of the ephrin, one of the ephrins called A5. It's very high in caudal superior colliculus. Later in development, at P8, it starts to decline, but still higher caudal. And then by 2-3 weeks of age, it's kind of going away because it's not really needed there anymore, so perhaps it's not produced. For some reason, it's downregulated. That's an interesting question in itself, but we don't know the answer. So to set up the hypothesis then, what we're testing with this descriptive study, we're going to look at how the expression pattern of the ephrins in the target changes as a result of removing part of the target structure. And there's two possibilities, right? If we just cut off the gradient right there, then we would get this situation. We'd go from yellow to pink, but not all the way to red. If the gradient compresses, which was our hypothesis, because we're thinking the compression of this gradient would cause the projection from the retina to compress on the smaller fragment, then we would see the slope of the gradient get steeper. So it would go all the way from yellow to red, but in a smaller distance. And this is kind of an ugly figure, but that's what we found. So here's a normal superior colliculus. You see the high ephrin label there. This one is, I had a different mood that day, so it was blue and green instead of red and yellow. And here we have our smaller tectum. Superior colliculus was lesioned on the day of birth. These are now P5 animals, I believe. And we see that the high ephrin expression is still there, but that peak has moved forward, still at the back of the superior colliculus, but the superior colliculus is smaller in these damaged tectum animals. And you maybe see from the arrows here the front and the back of the tectum. So it's roughly, we try to make roughly 50% lesions. You can actually remove about 70% of the superior colliculus, and you wouldn't know it from the way the animals behave. And in fact, the system compensates with receptive field size up until about that point. You go beyond that, and it starts to lose it. But then it loses it in a really intelligent way. It starts to represent only things here and sacrifices what's there. You remember that these guys have their eyes on the side of their head, so their visual field is like this. So gradients are steeper. And just to give you an idea again about the hypothesis, so if this is our normal map steepness, so looking at the levels of ephrin-A5, and this is just a cartoon, it's not meant to be real, but opposed to SC location, superior colliculus location, from the front to the back, it's about 2 millimeters in length. In the normal animals, if this is our slope of the gradient, if we had just cut off the gradient and reduced the size of the tectum by half from 2 millimeters to 1 millimeter, and nothing changed, we would expect the gradient to look like that. It would never get up to that high level. If our hypothesis was correct, then we would expect the gradient to get steeper. So it would go all the way up to the same level, but in 1 millimeter instead of 2. And that is, in fact, what we found. So here are plotted the slopes across development. Normal animals in black, the damaged tectum in white. And this is the slope, so the change in the mRNA expression over the map location. We get steeper at each age in the lesion animals. So that supports the hypothesis that the change in configuration of the molecular signals can direct the compression of the retina onto the smaller target but it's only a correlation. So this is where I got dragged kicking and screaming into using mice. And when I say I mean the royal I. So my very talented team was able to develop the mouse recording. We had to make a number of modifications because they're a little bit smaller at all stages of life, but it wasn't too bad. So I already told you that. So just to remind you then, the result of the correlative experiment was that the ephrin gradient goes from yellow to red, but in the smaller distance. And we also found, I'm not going to show you the data because I don't want to keep you here forever, but the gradient of receptor seems to compress in response. So this happens about a day later than the compression of the ligand gradient. So we see the receptor gradient changing. And I say about a day later, we only looked at 24-hour periods. So for all I know, it only takes an hour, but we need to run that down. That's one of the things we're doing now. Okay, so now we want to know whether those ephrins are causal for MAP compression. And so we have to knock them out. So if the change in ephrin expression is necessary for MAP compression, then if we knock out the ephrins, it should be impossible for the gradient to compress. So we worked in knockout animals. That's why there's a little mouse at the top there now. And we did the same electrophysiology experiment. In the normal animals, of course, we had that data already. We took the, although not in mice, so we had to repeat the entire thing in mice, normal mice. We then took the normal mice and ablated the back half of the tectum, same as we had done in the hamsters. And indeed, this was pretty fortunate. They also compressed their MAPs. It's not just a hamster thing. And that's important because mice take longer to develop. So even though we're looking at an animal that takes more than 15 and a half days to hatch, we still can get that compression. So it's still early enough in the development of that optic nerve growth to do that. And so what we're hypothesizing is that in the ephrin-A knockout animals, that instead of getting this compression, they aren't going to have any ephrin, so that's why this isn't yellow or pink anymore. We're not going to get the compression, and we'll still only map half of the tectum. So here we can go a millimeter with our recording electrode and see the entire projection smooshed onto that target. We're proposing that in the knockout animals, we're just basically whacking off the map in half, and we're only representing half of the visual field. And so here's where we get into these really complicated pictures that I've never presented to an audience before, so we'll see how this goes. We are working with a rather incredibly famous computational neuroscientist by the name of David Wilshaw in Scotland. We have all these computational people in town, including many of them at my own institution, and nobody's interested in sensory systems. No, I don't mind going to Scotland. It's a lovely place. But the thing about David is he has been fascinated with this question of retinotactile mapping all of his life. So I called him up like, you don't know me, but I think you could really help me with this. He's like, yes. So we actually met at the European Neuroscience meeting in Copenhagen, and he developed a way for us to statistically analyze and to visualize the form of the map. So let me orient you here. So these are just normal animals, wild type mice. I think this one you can see pretty well. So as we move our electrode across the tectum, from front to back and from medial to lateral, we get nice even progressions of visual field location. So we see how in this map of the visual world, the tactile locations are very ordered, right? There's a very neat topographic projection. We can also represent that, and we know that the same thing happens in the animals with ablations, right? Even though the tectum is half as big, we compress the map, so we still get entire visual field represented on that smaller space. So these graphs just show our recording locations. So we made 3 series of penetrations along the anteroposterior axis of the superior colliculus. And these maps are how David represented actual visual field location. And you'll see that these maps are mostly blue with occasional little red lines. The red lines mean that there's some topographic messiness there. So in the normal maps, they look pretty good most of the time. They have very little red, and there's a very, it's not linear, but it's biology. Why would it be linear? So it's a nice representation of the visual world, a little expanded in the front. In the reduced tecta, as I said, we know that the map compresses, so we would expect it to look roughly the same, except that our electrode positions are going to end where the lesion started. So this one was about not quite half, 1 and a little bit millimeter, and we try to shoot for 1, but it's difficult to get it exactly right. But again, you see that these maps look quite normal. If anything, even less disorder in that map, which is interesting. Now, we had a number of options to pick from in terms of the mice that we could use. How much time do I have?
10 minutes.
Okay. That's good. So we initially got these super duper everything knocked out mice from David Feldheim at UC Santa Cruz. And just, for the students' edification, I was asking for these knockout mice that David Feldheim developed. And at the time, his house was being bombed by animal rights activists. And they had to throw their child out the window to escape. And he was injured. His wife and child were not. But that slowed down the mouse shipment a little bit. So eventually we got these mice, and they're shipped as, there's several different ephrin-As. So they're shipped as A2, A3 knocked out. A3, as far as we know, isn't involved in the topography. A2 is a little bit involved in the topography. A5 is the major one, and they're heterozygous for A5. And so if we cross them, we can get wild type A5, heterozygous A5, or a triple knockout. And the wild type A5 then would be double knockout. So they're A2, A3 knockout, but A5 is still supposedly okay. And you'll see that the results look quite different. So it looks like we're not able to project very far with our recording electrode, even though we're going the same distance. We're not able to represent all the visual field locations. Now you're familiar with these drawings. You see, oh, I forgot to put that one on there. You see that they're pretty messed up, even though A5 is wild type in these guys. And it looks like they're certainly having trouble mapping the back half of the tectum, or even the front half in some cases. There's a lot of variability, so we don't quite know what to make of that yet. And these are just the normal guys, right? So what about the reduced tectum ones? We see in particular that they're not mapping for the most part. There they get a little bit of representation in temporal retina, but mostly they're only making half a map. So we expected them to be messed up in topography. That was already known because these animals were used for topographic mapping. That's why they were created. But no one had ever studied the role of ephrins in the compression, the topographic map plasticity. So these double knockouts are kind of interesting. So then we got another strain where it was just A5 knocked out, hoping to get a little more consistent picture. And we got these from Renping Zhu at Rutgers, who wasn't firebombed, and so we got them right away. Here's the normal, not lesioned animals with no ephrin-A5. The topography can be a disaster, or it can be fairly normal. Interesting, the variability. If we reduce the size of the tectum, so we lesion the back half of the tectum at birth, these are recording sets, so you see we don't get to go as far because there's not as much tectum before I run off the end. With these guys, we occasionally get mapping of the back of the tectum, and sometimes we don't. But the topography is quite variable in how messed up it is. So this is why I didn't want to work on knockout mice. Now the triple knockouts. I have to be equal opportunity. So the triple knockouts, as we expected, their topographic maps are a disaster. I mean, you can put your electrode at the front of the tectum, you get a field here, you move it just a tiny bit, 200 microns, and you're back there already. Move it a little bit more, and you're here, right? So it's just really crazy. So if you look at David's plots here, you see how the topography is a complete disaster. And we're really unable to even know whether we get a compression of the map or not, because there's no map. So those animals weren't especially useful. And so we were unable to calculate a map slope with the triple knockouts because they were just too crazy. There really was no map. And we're thinking, well, the axons, because they don't know where they are, they have no idea that there's some part of the tectum that's missing. So they just go wherever. And the entire retina might go to the target, but there's no way for it to distinguish which part of the target it should end up in because it doesn't have the signals that tell it to get there. What David is most interested in is that you can get little bits of orderliness in these projections. And he's wondering, are those the axons that are special? Maybe there's a special kind, because of course there's lots of different kind of retinal axons, and maybe there's different retinal ganglion cells that are more successful in surviving this sort of manipulation. But so with the absence of the TKOs where we could not map the slope, we see that the slope of the map, so here's our regular guys, right? So the slope of the map is about here. In the wild type animals that have the ablation, the slope has to get steeper to get all the retina represented there. But if we do the statistics, we see that in every one of the knockouts, excepting the TKOs where we can't calculate a slope of the map because there's no map, they are having a problem, some more than others, with compressing that map and increasing the slope of the projection. And I haven't done the statistics yet. That's up for the SFN meeting, so I couldn't quite get it done for you guys, but it's going to be interesting. So if we actually look at the slopes, here's our normal animals with all of the data, and there's our lesioned animals wild type, so much steeper. There they are here, and the different genotypes are mapping somewhere in the middle there. We thought, well, that could be due to the fact that we didn't lesion them all the same, but the groups don't have distinguishably different lesion sizes. So they're all roughly half. Okay, so that supports the causal connection then, that we do interfere with compression of the map if we don't have the ephrins. It's definitely not clean data, so we're going to have fun trying to sort out exactly what's going on, and that suggests a lot of further experiments. But just to summarize then, I've told you how activity-dependent mechanisms through NMDA receptors can compensate and preserve stimulus tuning, so response properties. The compression requires at least ephrin-A's and maybe some other factor that help get some normal topography. But if we knock out those genes, then we don't get the compression. So we have defined then now a number of different factors that allow us to understand what mechanisms are used by this very simple projection to preserve function in the face of rather challenging damages, removing half of the structure. So just to acknowledge all the different people that were involved, then I don't think I can reach that high, but we have our many collaborators here. I didn't get to talk about it, but Josh Sains has just sent us some transgenic mice that have different populations of ganglion cells labeled with GFP, so we can start to look at differences between types of ganglion cells and how well they survive this manipulation. Tizita did the correlation study. Yu-Ting was the beast who not only did ferret recordings, but pioneered the map recordings as well. Tim Ballmer was working with her. He's now a postdoc in Larry Trussell's lab at OHSU. David Mudd is my current student, and then a number of different postdocs, and my very first student, Magdalena Carrasco, who was not involved in these projects, but I'll mention her anyway. We now have funding from NSF. We did have funding from NIH and a number of other organizations. The Brains and Behavior Program and the Center for Behavioral Neuroscience at Georgia State provides seed money for us to do this work when we can't get any funding. But now we have some funding, so we get to proceed. And with that, I'll finish and take any questions that you have. Thank you.
Great talk.
Thank you.
First of all, and I'm sorry if you said this at some point, but did you see any changes in upstream structures other than where they project?
So there has been one group that looked at the projection from cortex back to tectum in these knockouts, but not in a lesion scenario.
And did you see any changes in retina that?
We didn't record from the retina, so we don't know, but that's a really interesting question. It's a little challenging to record from the retina in a mammal. You can routinely do it in frogs, for example, but it's difficult in a mammal. Sorry. Yeah?
Do you know which cells express the ephrin-A in the tectum? And how would it change the
We don't know exactly. It seems to be expressed not only in the tectal target cells themselves, but in the extracellular matrix, and that may be because it's secreted, but there hasn't been a lot of biology done on that in the visual system. Now, there's been a lot of work done on it in cancer systems, but I don't dare imagine that it would be the same. Good question. Yes? They were all anesthetized. Would love to find a student who wants to do the work to develop the awake behaving prep.
Do you have any theories?
So the way that we're kind of lucky compared to people who are working in monkeys or carnivores in that in the rodents we can use an anesthetic called urethane, which we think of as a plastic wrap, right? But it's also an anesthetic. And it suppresses all the different neurotransmitter systems fairly evenly, so we don't have the same problems like we would with ketamine of selectively depressing NMDA receptors. But still, it would be much better to do this in an awake preparation. Thanks for that question. Others?
One question.
Mhm.
Do you have any idea if the excitatory inhibitory cells are affected differently?
I have no idea because we have not recorded from the inhibitory neurons directly. We would have to do that in slices. There's been plenty of recording from the very large inhibitory neurons in cerebral cortex, as I'm sure you know. But in superior colliculus, I don't know that there's been very many people that in slices have recorded from the inhibitory neurons. And that's something that I really would like to do because I'd like to see, I mean, even just to look at them morphologically, how they're changing in this scenario. They should be changing morphologically, and that would be something that's fairly easy to address. We just haven't done it. Thanks, that's a good question. Yeah?
Do you have any sense, I think you said that receptor fields in superior colliculus were fairly small.
Well, it depends what you compare it to. Yeah. They're much bigger than visual cortex, but they're small enough that we call them bug detectors.
If you move to higher areas, higher cortical areas that are typically studied
Mhm.
that big receptor field, what's your prediction?
Well, in V1 and V2, they'd be smaller, but as we go into the dorsal stream, they would get larger. Yeah.
So, I mean, but do you think there's a limit to where the compression is really intriguing?
Mhm.
Because I think I would predict the same thing, but then in these areas that have massive receptor fields as you move in the dorsal stream, would that break, do you think?
So we don't know because we haven't recorded there, but judging just what happens in the tectum, you can break the system by removing about 3/4 of the target, but not before. So presumably all that compressed information is being piped up through the dorsal stream and along the ventral stream to operate in stimulus localization and object recognition, such as it is in a hamster.
Okay, thanks, Sarah, for a great talk.